Okay, good morning, everyone. We're going to make a start. Just to introduce myself, my name is Dr. Maurice Gleeson. I've been involved in genetic genealogy now for about 5 years or so, and I'm a member of ICARA, the International Society of Genetic Genealogy. I have a surname project, the Spierin surname project, that uses yDNA to trace our Spierin line. We've managed to get back to the 1600s in Limerick. And I also organised the first genetic genealogy conference in Ireland last year in October. And the good news is that Family Tree DNA, who sponsored it last year, will be sponsoring it again this year. So if you're in Dublin in October, then please do come along to Genetic Genealogy Ireland 2014. So this morning I'm going to talk to you about autosomal DNA, a step-by-step approach to analysing your autosomal DNA matches. Now, let's look at, first of all, what we're going to cover. First of all, what is our goal? We'll have a look at that. What is autosomal DNA? What is a match? And then we'll look at a step-by-step approach to analysing your matches, and then we'll look at future directions for autosomal DNA. But can I just ask the audience, first of all, how many people have had their autosomal DNA tested? All right, great. So there's quite a few people. Fine. So you'll be familiar with a lot of the stuff that I'm going to talk about. For those of you who haven't had your autosomal DNA tested yet, don't worry too much. This just is a little bit over your head sometimes. I'm going to try and keep it as basic as possible. We are recording all of these sessions. We are setting up a YouTube channel, and hopefully the audio is going to be good enough that it'll actually be understandable. Can you actually hear me okay at the back? Okay, great, fine. So, what is our goal? First goal is to confirm known or suspected relationships. Secondly, we can use autosomal DNA to connect with distant cousins. Thirdly, we can use what they know about their family tree to break through the brick walls that we have in ours. And fourthly, to a certain extent, it's to determine our ethnic makeup. Now, that can be useful. You know, if you're Irish like I am, you're not going to expect anything more than you're Irish. I knew that already. But if you are, say, American, where there's a large melting pot of different races and different ethnic groups, then this can be very, very useful in helping you decide which ancestral line your match belongs to. So, let's start off then by just looking at what a brick wall is. And here is my family tree. You can see me there at the bottom, my parents, grandparents, and I've traced my family back to about 1800 for about half of my ancestors, and about 1835 for about the other half. And that's after exhausting all of the usual traditional documentary records. In Ireland we have Griffith's valuation, and the valuation books, the 1901-1911 census, church records, birth, marriages, deaths, and then uploading my tree onto the internet and collaborating online with people, and also looking at family lore, newspapers, Glasnevin cemetery records, electoral rolls. But there is my brick wall. And in order to break through my brick wall, I've had quite a bit of luck with yDNA. And using that in my Spierin surname project, we've been able to piggyback and jump back to the early 1700s, the late 1600s, with that particular ancestral line of my family. So DNA testing can actually help you break through some of those brick walls. But what is autosomal DNA? Well, there are three types of DNA. The yDNA is passed on from father to son. So if you did a yDNA test, it would take you back along your father's line, all the way back to about 338,000 years ago in Africa, when we were all, we didn't have the floods that we have today. So that's the yDNA. On the far side, of the other side of the tree, we have the mitochondrial DNA, which is passed on from a woman to all of her children, but is only passed on by her daughters. So the mitochondrial DNA will take you back along your mother's 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 line, all the way back 200,000 years ago to Africa. Now, it's the third type of DNA then is the autosomal DNA, and that looks at all of your ancestral lines between the both extremes of your family tree. And that's what we're going to talk about today. Now, I've had my autosomal DNA tested, and that gives me a reach of about 7 generations. And that's the limitation of autosomal DNA compared to Y and mitochondrial. Y and mitochondrial are good for deep ancestry as well as recent ancestry. Autosomal DNA is only good for recent ancestry, say within the last 200 to 250 years. You get a match with somebody on your autosomal DNA, it implies that you share a common ancestor sometime in the last 200 to 250 years, approximately. Now, I've also tested my dad, so I've pushed back my reach on my father's side by an extra generation to maybe a common ancestor that was born around about 1730, and I've also tested my maternal aunt, so I've also extended the reach on my maternal side to about 1730. But let's take a closer look at the DNA sample that you might give if you went over to Family Tree DNA and had your cheek swabbed. And they'll swab your cheek, put the end of the swab into a little test tube and send it off to the lab. But what you'll find is that in that swab, because in that test tube, because you've been swabbing your cheek, you've dislodged some of your cheek cells, and inside your cheek cell, you see it here, you have these little blue things which are called mitochondria, and that's where you get your mitochondrial DNA from. But in the nucleus of the cell, you have your 46 chromosomes, organized into 23 chromosome pairs. And the last of these pairs, because you've got one copy of each chromosome in each pair, the last of these pairs are called the sex chromosomes. Now, all of the other ones are virtually the same length, same kind of shape and morphology, but the last ones, the X and the Y chromosomes, they're different. In women, they've got 2 X chromosomes, which makes them women, and in men, they have an X and a Y chromosome, which makes them men. The difference between the X and the Y chromosome is that the Y chromosome is only about 1/3 of the length of the X chromosome. And so your average man is less of a person than your average woman. And they've proven it scientifically. Okay? So, let's take a quick look at how autosomal DNA is passed on. And there's 2 important processes in operation. The first is independent assortment, and that basically means that because we get half of our chromosomes from our father and the other half from our mother, 1 chromosome in each pair we get from our father, the other chromosome in each pair we get from our mother. So what happens when, during the formation of egg cells and sperm cells, all the chromosomes line up, and then half of them go to one egg cell, and the other half go to another egg cell. But it could be, you don't know which one is going to go to which cell. So what you get from your father, you only get 50% of what he has. What you get from your mother, you only get 50% of what she has. So with every generation, there is a halving of the chromosomal load that is passed on. The other half is completely lost to you. So you have no access to the half of your father's chromosomes that you didn't get. You have no access to the half of your mother's chromosomes that you didn't get. That's the first process. The second process is called recombination, because it's not as simple as just getting one chromosome from your father. Before he actually passes on whichever one he chooses to give you, the 2 chromosomes recombine and cross over, and they swap little segments with each other. So over time, the DNA in the chromosome, the segments, it gets segmented. And that's why when you look at your matches, you'll find these DNA segments. In essence, your father, your parents give you grandpa with a bit of grandma, or alternatively grandma with a bit of grandpa. Okay? So those 2 processes result in what is really a genetic lottery. How much DNA do you get from each of your ancestors? Well, if we look at a 4 times great-grandmother, and for me that would maybe be somebody born in about 1765, she passes on 50% of her DNA to each of her children. But if you look how it's passed down through the generations, the contribution from your 4 times great-grandmother gets halved with every single generation. So for me, born in around about 1960, my DNA is only 1.5% of my DNA I got from my 4 times great-grandmother. If you look at how much DNA you share with your relatives, that gets halved too with each generation. So that siblings only share 50%, first cousins 12.5%, and by the time I get down to my 5th cousins, 0.05% of your DNA is shared. That means that the test is less able to pick up that DNA as the further the relationship is. So at the level of 5th cousins, you only detect about 10% of your 5th cousins. Now, the important thing to remember, though, is that you're going to have a lot more 5th cousins than you will do 4th cousins or 3rd cousins or 2nd cousins. Because if you allow 3 children per generation per cousin, you can see that when you get down to 5th cousins, you've got 16,000 5th cousins. And if you're only detecting 10% of them, it's still 1,600, which is a hell of a lot of people. So when you do the test, you'll find that most of your matches are distant. Less of your matches are going to be close, 2nd or 3rd cousins. But let's go on a fishing trip, because that's what most people do when they take a family finder test with family tree DNA, for example. You're just putting your data into the data pool and seeing what you get. Now, there are several companies that offer an autosomal DNA test. Family Tree DNA is here. All of the companies offer it at $99. The trouble is, with 23andMe, you have to add an extra $80 on for postage and packaging, because they send it by DHL. And for Ancestry DNA, it's not available outside the US, so you're going to have to hop on a plane, fly to John F. Kennedy Airport, go to my friend Thomas's apartment in Manhattan. He's going to be willing to admit you can swab yourself there, hop back in the taxi, and then get the overnight flight back to London, and that's going to cost you £630. So there is a little bit of a differential between the tests. Now, no matter who you test with, do upload your results to GetMatch. It is completely free. It is run by 2 volunteers, and that's why it's so slow, but it is completely free, and this will allow you to compare your results with anybody else who's uploaded their results there. Not just from Family Tree DNA, if you test with them, but from 23andMe and from Ancestry DNA from the other companies. So do upload your results to GetMatch no matter who you test with. Now, these are my results on Family Tree DNA, and if you were here for Debbie's presentation previously, you'll recognize this. We have all of my matches. I've got somebody called Hare, somebody called Haddon, a Spierin, a Swords, a McGuire, and it tells me various things. It tells me my match date, my relationship range, my known relationship, if there is one, a shared centimorgans, if there is one, shared centimorgans, and total, the longest block, 38 centimorgans. So that's the kind of results you get when you get your family tree DNA. And those are all my matches. But what is a match? A match is someone who has an identical segment of DNA compared to you. And that implies that somewhere along the line you have a common ancestor. Both of you are descended from the same person, or, because that common ancestor will be married to Mrs. Common Ancestor, the same ancestral couple. Now, let's take a quick look at your average chromosome. So let's take chromosome 21. And this is just to explain how the test works and what they're actually comparing. This is, if you unravel the chromosome, you see your typical double helix, the 2 strands of the double helix. And you have all of the bases down here. Now, these bases are guanine, cytosine, adenine, and thymine. You don't really need to know a huge amount about this, so I'm going to run through it very quickly. Guanine only combines with cytosine, and adenine only combines with thymine. So you'll only see G and C combining together, and A and T combining together. So this lower strand of the chromosome here is an exact mirror image of this strand here. A, T, T, A, G, C. And because of that, we don't actually need to read both strands of the double helix. We actually would be able to know exactly what the other strand is because it's a mirror image. So technically speaking, we only read one strand of the double helix for any given chromosome. But of course, we're not just comparing one chromosome, we're comparing two chromosomes with two chromosomes. Because the test, the machine that they use for analyzing your matches, all it knows is chromosome 21. Oh, well, there's two of them, so I'll give you two results. It can't tell you which one you got from your mother and which one you got from your father. And the result it will give you is, if I say to it, what's the reading at position 1? It will say, well, you've got a T and you've got a T. And at position 2, well, you've got a G and a G. At position 3, you've got an A and a G. One of those is going to be on one chromosome and the other one will be on the other chromosome. And when you come to looking at your match, it'll do exactly the same. It will read what's on your match's chromosomes. And you'll see it's going to compare your results with your match's results and give you, it'll say whether it's completely different, it's fully identical, whether it's half identical, you only share one letter in common, or whether there's no letter shared in common at all. And that's what we get here. Non-identical regions, fully identical regions, and half-identical regions. And if you upload your results to GetMatch, you'll probably be quite familiar with this type of diagram. And the red regions are non-identical regions. That means your two bases don't match any of the other two bases in that particular position. The same goes for fully identical region. You've got an exact match. Both chromosomes match the other position of the other two chromosomes of your match. And then the half-identical regions in yellow just indicate that one of your two bases matches one of the other person's two bases. So that's just to give you a brief overview of what you'll see on GetMatch and what we're actually comparing. It means also that you can get false matches. When is a match not a match? When it's a false positive. And if this is what you're comparing to, this is what you've inherited from, say, your 4 times great-grandmother, you can get a false result if the machine reads these bases here. To the machine, it's going to look like this. So it's going to say, oh yeah, that's definitely a match. Much more commonly what happens is you get a true positive with a little false positive at each end. And that's called a fuzzy segment. Alternatively, you can get a compound segment where you get a true positive on one end combined with a true positive, but it isn't exactly the same as the one that you've inherited from your ancestor. Now, the implications of that are that there are going to be a certain amount of false positives in your results. How do you recognize them? And they've done an analysis, if your segment is 8 centimorgans or less, you've got a 50-50 chance of it being a false positive. It's better to go for a 10 centimorgan match or greater because you've got a 99% chance of it being identical by descent, or in other words, a true positive. And if you go down to a match that's only 4 centimorgans in size, that is going to be a 99% chance of having a false positive. So, that brings us to the step-by-step approach. And the first step is to determine where does your common ancestor sit. The second step is the common ancestor obvious? The third step is to eliminate non-contenders, and the fourth step is to work with small related groups. Now, the great news is, all that technical stuff I just showed you, you don't need to know any of that for the first 2 steps. So, no matter what your level of knowledge about the science behind DNA, you don't actually need to know the intricacies to actually get some very, very useful results out of it. We're going to focus on several different tactics. We're going to use DNA sometimes. We're going to use genealogy, just traditional genealogy other times. And we're going to do group work in the final step. So let's look at the secrets to success. Start with your closest matches. Share your tree. This is really, really important. If you don't share your tree, you've got a very limited chance of actually finding where you connect. Develop a relationship with your matches. it's important because if you just send them a quick email, do you have this, and you don't work on developing a kind of a nice friendly relationship with you, you're just going to get a yes/no answer, and that's the end of it. And also you need to keep track of what you've done. So, step one, where does the ancestor sit? And this is just really about positioning the common ancestor on your family tree. And this uses just traditional genealogy. So, for example, if the results come back and says, we're third cousins, you and your match are third cousins, that means you share a common 2 times great-grandparent. And for me, that would mean that he was born around about 1835. Okay, so now we know where we want to look. In terms of my 2 times great-grandparents, I've got 16 of them. But each of them is married to one of my other great-grandparents. So there's 8 couples. So all I need to do is compare my 8 couples with my match's 8 couples and see if 2 of them are exactly the same. Similarly for 5th cousins, if my match comes back and says, we're probably a 5th cousin, then I've got 64 great-grandparents in 32 grandparental couples, and I need to compare my 32 4 times great-grandparents with their 32 4 times great-grandparental couples. So the problem was that the further you get out, the less accurate the estimate is going to be. So for example, if the test comes back and says, you're probably first cousins, then about 97% of the time that would be true. But if you get down to say 4th cousins, or let's say 3rd cousins, it says, and it predicts a 3rd cousin, it would predict a 3rd cousin maybe 70% of the time, but in 10% of cases it might be closer than a 3rd cousin, and in 20% of cases it might be further out than a 3rd cousin. And it's the same it's even worse with 4th cousins and worse with 5th cousins. So you have to be careful that when you get down to the 4th cousin, 5th cousin level, the relationship might be even further back than you imagine. It may not be at your 4 times great-grandparental level, it may be at your 5 times or 6 times great-grandparental level. Also, one thing that makes the connection seem much closer than it is if you have a lot of cousins intermarrying in your family tree, because more DNA persists than is usual. And this is called pedigree collapse. And you see this especially in some of the royal families of Europe. Is anybody familiar with Carlos II? Okay, well, do a Wikipedia or a Google search for Carlos II. He suffered as a result of, I think, about 3 cousins and an uncle and a niece marrying in very close proximity to each other. And this might be more relevant in small isolated communities like Ireland, for example, or the Orkneys, the Shetlands. And if there's endogamous populations like the Ashkenazi Jewish population, huge amount of intermarrying, and therefore relationships seem much closer than they are because the DNA has persisted in their particular population for a long time. Romani people, Hindu castes, Jehovah's Witnesses, these are endogamous populations where this may be a problem. The second step is to see if the common ancestor is obvious. And in order to do this, you really have to characterize your brick wall. It's fine if you've got a family tree that goes back to the 1400s and your match is a family tree that goes back to the 1400s. You'll be able to say, okay, well, we're just looking back far enough. Oh, yeah, there he is. There's the common ancestor. But if you've got a brick wall, you really have to characterize that brick wall in order to make the most of anybody that you match. And that means identifying your most distant known ancestor on each of your ancestral lines, looking for BMD records. Maybe there's an address in the birth, marriage, and death records that they have for their children. So can you identify a location? What about witnesses to baptisms, witnesses to weddings? Usually it's brothers and sisters that are best man, maid of honor, godmother, godfather. Look at the witnesses in your most distant known ancestors' children baptism records to see if you can identify likely candidates for either a married sister or a single sister for the most distant known ancestor, or for anybody else in the extended family. Also locations as well. These are very, very important. So when you come to matching with somebody and they say, we've got a brick wall here, and you say, my brick wall is here, you're going to have to look for these clues to try and see where your traditional genealogical research is going to lead you next. Now, the problem with this is, where do you put this information in your tree? And one of my problems at the moment is that I don't know where to put all the sponsors and godmothers and godparents from my most distant known ancestors' children's baptisms in my tree on Ancestry, so that other people can see it and go, oh, your godmother of that person is my sister of my most distant known ancestor. So that's a problem. I haven't overcome that as yet. If you test with family tree DNA, this little symbol here indicates that your match has uploaded their tree onto the database. So you'll be able to click on that and you'll be able to see their tree. That's great. On the other side, you'll see that there's room for entering surnames. You know, it's a substitute for a tree. It's not the best thing, but it is a good thing to have. And in my dad's results, he's got 355 matches on Family Finder. 66% of his matches have got surnames in their profile, but only 20% of them have uploaded a tree. So if you can't access the tree or you can't access the surnames, then you have to contact the match. But there is another way. If there's no tree, you could try to find one. And I've had a lot of luck with this, actually looking for trees online. Now, here's my profile. If you match me, this is the kind of result you're going to get. But if you click on my name, what will come up is my profile. And it gives you my name, it gives you my email address, and it also gives you my most distant known ancestors. John Gleeson with his date of birth and death, and Catherine Farrell. So you can use these to do a Google search and to search online on the various online family tree groups like Ancestry, MyHeritage, Luke's Web, WikiTree, etc. And with a bit of luck, you'll actually find my tree there. So if that doesn't work, then you can always contact your match. The problem here is, some of them don't reply. Some of them won't share their details with you. Some only give partial information, and a lot of the time, many don't have an up-to-date tree. You know, oh, I just started and I decided to do DNA. Can you give me any hints and tips? So it can be difficult, and if you don't have the tree, it makes it really difficult for you guys to actually find out who that common ancestor is. Step 3 in the process is to eliminate non-contenders, and we're going to look at a variety of different techniques here, about 7 altogether. And we're going to use DNA. This is the first time we start using DNA, as well as traditional genealogy. Now, the goal here is to get rid of your ancestors. Okay, so it's like a card game, it's like whist or poker, and you want to whittle down your ancestors. Is it maternal or paternal? Get rid of 50% of them. Fantastic. Is it a match on the X chromosome? You can get rid of even more. Is it an improbable ethnicity? I've got Irish on one side, Japanese on the other side. That's a lucky ethnic group to have. And what about grandparents? You know, you might be able to identify which grandparent your match lies on. So these are the various techniques we're going to look at. Now, myself and my dad have tested. If a person matches me and my dad, then I'm fairly convinced that it's going to be on my dad's side. It's not on my mother's side. But the opposite is also true. If a person matches me but does not match my dad, then the match must be on my maternal side. Okay, so that's really simple. That's really straightforward. What about testing other more distant family members like cousins? And this is an example of my tree. And there I am down there. There's my siblings. I've had them tested. There's my dad. There's his sister. There's my maternal aunt. There's my dad's second cousin. There's my dad's second cousin once removed. There's somebody else down there as well. So there's actually, I've tested quite a few people. And if it turns out that I have a match with somebody, and then I look to see if they match anybody else, and oh, they match my dad's second cousin. Well, that means, because I know that my dad and his second cousin are connected via my great-grandparents John and Anne Gleeson, it means that this match is probably connected to us by a common ancestor at or above the level of those particular great-grandparents. So that really narrows down my search. So testing cousins first cousins, second cousins, to focus the research on a particular ancestral couple in your line can be a very useful technique. I mean, it gets costly, obviously, because they'll say, sure, I'd love to test. When can you send me it? So, another thing just to mention briefly, if you've had your Y-DNA tested, and a lot of people who've had Family Finder have the Y-DNA test done as well, we know that comes down the father's line, father, 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 father. So if you match somebody on your Y-DNA, that means you've got a genetic connection with them that goes up father's 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 line to the common male ancestor, and then down father's son, 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 son to them. So the two direct male lines converge. If you have a match on your Y-DNA, then that means that you are possibly connected via that particular direct male line up and direct male line down. Now, you should know that already, because if you share the same surname, you should have a match on the Y-DNA. If you don't have a match on the Y-DNA, you still could be related by Mrs. Common Ancestor up here, and you could still be related via that couple, but the DNA that you got was from the wife and not from the man. Similarly, on the mitochondrial DNA side, we know that the mitochondria, if we have a match on the mitochondrial DNA, it implies that we are connected perhaps via the direct female line going up to the common ancestor and the direct female line coming down. If there is no match on the mitochondrial DNA, it implies that the connection cannot be via that direct female line up and direct female line down. It still could be connected by the husband of that particular ancestor, but it's just another way of, if you've done the mitochondrial DNA test, look at it and see if there is or is not a match. It will give you some information. atDNA is has fascinating transmission, and it is unique transmission, because if you're a man, you get the X from your mother and you get the Y from your father. But the X DNA stops at your father. You cannot get it from your father. So if you're a male and you've got a match on your X chromosome, it can only have come from your maternal side. So immediately you've eliminated 50% of your ancestors, and that's a fantastic result. If we look at the path of transmission of the X chromosome, you can see that it's very heavily weighted towards the female side, going all the way back. And in terms of eliminating ancestors, the further you go back, the more ancestors you can eliminate. So that you have a very good match on the X, you know it can only come via these ancestral lines. Now, I've drawn a chart that shows you my ancestral lines with all of my ancestors put in them. If I have a match on my X, I know it can only have come from one of these ancestral lines, and that is a fantastic way of narrowing down which ancestor is the probable common ancestor. And your match could do the same kind of chart. If your match is a male, then you'll get this kind of chart. If your match is a female, then you'll get this kind of chart. So if you're a female, it doesn't outrule the male side, but it still does cut down the number of ancestral lines. Now, in terms of ethnicity, nationality, and geography, if, for example, you have a family tree that includes Irish, Japanese, African, and English, then that's a very easy way of ruling out a particular ancestral lines. So, for example, if your match comes back and says, oh, well, my family's Japanese as far back as 1200, then you know it has to be only in those ancestral lines that are Japanese. And the other thing about that is there are certain tools on GEDmatch, if you upload your data, where you can do a chromosome by chromosome breakdown of your ethnicity, which can be very, very useful for telling which of your chromosomes you inherited from which ethnic group in your background. This is also a very useful technique, but very time-consuming, and you will need to test first cousins, second cousins, third cousins. It's Whitaffy's technique. He uses a technique where you either have two parents or one parent and several siblings, and by looking at their DNA, you can figure out which grandparent each part of your chromosome came from, which segments you got from each of your grandparents. And that can be quite useful as well. But like I said, that will take you weeks and months of work. But at the end of step 3, what you'll find is that your hand of ancestral cards has been whittled down. And it really can help you confine your research to just 1 or 2 ancestral lines if you're lucky. If your match does exactly the same, then it really does help focus your research. But brick walls may still persist. And that's where we come to step 4, which is working with small related groups. Now, I liken this to playing a game of cards and having people around for a game of cards. And the idea is to, first of all, assemble the people that should be sitting around the table, establishing who matches who, and then repeating steps 1 to 3 for each of those individuals, and comparing trees at each stage. If you cannot compare trees, you've got no hope of finding that common ancestor. So this is where we get into a little bit more involved activity. But if, for example, you imagine that these are all my matches, and rather than names, I'm using letters and numbers, and this person over here is my match, and she's got a whole list of matches as well. But when we compare our list of matches and look at the people who we actually have as matches, we find that we share exactly the same people in our list. So these people here and these people here are called our common matches. And the idea behind the common matches is that it is likely that somewhere in the past, all of us, me, my original match, and all of the matches that we share in common with each other are connected to each other by a common ancestor. That's the theory. But nature always likes throwing a fly in the ointment. And here is the fly. What are the chances that we're connected not just via one common ancestor, but by a second common ancestor? One common ancestor on the paternal side, and another common ancestor on the maternal side. Would anybody like to guess what is the probability of that happening? Shout out a number. 10%? 15%? 50%, somebody? Right, it's a trick question. The answer is 100%. If you go back far enough in time, you will find connections on both your maternal and paternal side to your match. Okay, if your match is Japanese and you're African, then you might have to go back several tens of thousands of years to find that connection. But you will find it. If, on the other hand, you come from a small, isolated community, like Ireland, for example, or the Shetland Islands, or Newfoundland, then the chances of you having a common ancestor, a second common ancestor, at a much more recent time is very, very high. So these common matches could either be on the maternal side or on the paternal side. We don't know. Is there a way of maybe refining it a little bit more? Yes, there is. What you do to get these common matches is you go to your results and you click on common matches, and this box comes up. You click on in common with, and it will produce a list of your common matches. Now, the first thing to do when looking at your common matches is to look at the amount of shared centimorgans you have, the amount of shared DNA, and looking at the longest segment. And you can see that there's my father. Our longest segment is 267 centimorgans. There's my brother, there's my sister, there's a potential third to fifth cousin. It comes down to 18. And here's one that only has 8.5. Now that 8.5 is below the threshold that I would normally accept for true positives, for false positives. So the first thing that I do is I put all of these matches that have less than 10 centimorgans, I put them to one side. Now there will be true positives in there, and I know in one case my brother ended up in that group. But it's maybe 50-50. Half of them are going to be true positives, half of them will be false positives. Put it to one side for now, we'll come back to that later on. You can also download results, which is also quite handy, and you can do spreadsheet work, which a lot of people do. The next step is to find out, with the remaining matches, do they match each other? And in order to do this, we use the matrix feature in Family Tree DNA. You go to your Family Finder tab, and you click on Matrix, and it brings up a box, and I've blurred out the names, but you can basically add the names that you had in that list of common matches. You can add them into the mix, and it will produce for you this type of matrix. And this has my dad and his first cousin. My first cousin matches my dad in common with all of these people, and by definition that's what common matches are. But if you look at my dad and number 3, he also matches number 5 and number 6. So 3, 5, and 6 all match each other. That means that they probably share the same single common ancestor. If you look at number 2, he doesn't match anybody in common. So he's kind of out on his own. He could be a match on the maternal side or on the paternal side, we don't know. If you go over to number 7, he matches number 8. Number 7 matches number 8. And that means that 7, 8, my dad and his first cousin, all are commonly matching each other, and they all probably share the same common ancestor. It's probably on the same side as 3, 5, and 6, assuming that there isn't a double relationship, or the second common ancestor is further back. So this gives you an idea of which of those common matches are more likely to be related to the same single common ancestor. And I like to draw a graph. That's because I'm the sort of person that I am. And there's my dad matching his first cousin, and I imagine in my own simple way that these are all of the card players sitting around the card table, and I'm able to map the relationship between each of these common matches all the way around the card table. Then I'm able to look at matches for 3 and 5, and 3 and 6. I'm also able to look at matches for 5 and 6, and I'm also able to look at matches for 7 and 8. So it's just another way of interpreting the matrix and putting it in a way that I find a little bit visually more attractive than the matrix itself. But you can see here that we're getting into an area where we're getting into a complicated area, really. Now, we can also check for overlapping segments. That's the other thing that you can do. If segments are overlapping, and you can see here that these people, 1, 2, 3, 5, and 6 are in this chromosome browser example, they have overlapping segments on chromosome 1. And because the segments are overlapping, there's even a higher chance that that particular segment was inherited by all of them from the same single common ancestor. And 2 of them overlap here, another 2 overlap down on chromosome 12, and another 2 of them overlap on chromosome 16. But it's the chromosome 1 which has person number 1, which is my dad's first cousin, and their common matches 3, 5, and 6, the green, the pink, and the yellow, that confirms or suggests very strongly that these people have got overlapping segments and therefore share a single common ancestor rather than 2. And one can do the same for the other matches. There's 7 and 8 on the other side, putting them into the chromosome browser. All of this can be done by a simple tool that was developed by DNA GetCom. If you haven't had a chance to go to DNA Adoption and look what the adoption community in the States is doing, please do, because they're developing some excellent tools. And this one from Don Worth combines the matrix and the overlapping segment facility in one lovely spreadsheet. So this is some great work that has been done by the adoption community, and it's something that you can experiment with, and it produces these lovely visual displays. So to kind of summarize step 4, and remember my dad had 355 matches. He has 26 matches with his first cousin. Of those 26 matches, only 7 of them have a segment that is greater than 10 centimeters, 10 centimorgans. 19 of them have segments that are less than 10 centimorgans, and these are potential false positives. So I'm putting them to one side. I'm not getting rid of them completely, but I'm putting them to one side. In terms of matching common matches, 5 of those 7 are matching common matches, and there's a greater probability, therefore, that they all share the same single common ancestor. And those same 5 have overlapping segments as well. And when I try and hone it even further by looking for a common location or a common surname among those common matches, 3 of them had a common location, but none of them had a common surname. So it's all a process of trying to narrow down, home in on the most likely people that you can share with to get information about your common ancestor. And the goal is overall to reconstruct a family tree for this small related group of common matches. Firstly, and this is just a recap on what we've talked about, you identify your common matches. You set aside those who have a less than 10 centimorgan DNA segment. You set aside those that don't have overlapping segments. And this allows you to focus on those who are likely to be related by a same single common ancestor. Then by doing multiple 3-way comparisons and triangulation, you can actually identify people within the group who are more closely identified to each other than they are to the rest of the group. And that can be very, very useful because you can tell those people, you guys are much more closely related to each other than the rest of the group. You're like third cousins, everybody else is fifth cousins. So you should really focus on your connection. And if we find out what common ancestral line you have, that's going to help us narrow down what common ancestral lines the rest of the group has. Compare trees at every single step. Eliminate non-contenders by using your maternal, paternal, other cousins, who matches who, by looking at the Y-DNA, the mitochondrial DNA, the X-DNA, and repeating. You could repeat the same process then with your kind of second-rate matches, the ones that you set aside, the ones that don't match the common matches, the ones that don't have overlapping segments, the ones that have centimorgans less than 10 centimorgan matches. And this will also indicate maybe further research. Maybe you need to do a Y-DNA on one of the particular lines. Maybe you need to do further autosomal DNA testing, or even mitochondrial DNA testing. So that is the step-by-step approach. It can be very involved, or it can be very simple. You don't need to go through each step in turn. You can pick and choose which steps you want and which suit your level of knowledge. But the secret to success is always start with your closest matches, share your tree, develop a relationship with your matches, and keep track of what you've done. Ladies and gentlemen, thank you very much for your kind attention. Now I have to check the time, and we have time for a couple of questions. Let me bring the microphone around. We have a question here in the front. I'll just give that to you.
When you went back, when you were first talking about GenMatch, there was something flashed up on the left-hand side about from a date, and then something else, and I've gone. What? Because I've done the
autosomal, yes.
Yeah. But I don't think I've done anything in GenMatch. So what was that thing that said from something in October of last year, I think? What was the...
No, it's okay. Let me go back through so it was at the very start of just talking about the autosomal DNA? Oh, right, okay. Let's go back to GenMatch then, which is just about there. That was the one. Oh yes, I see. Yes, if you're going to upload your autosomal DNA to GenMatch, thank you, Debbie, you will need to upload 2 files, your autosomal DNA and your xDNA. You can download each of them just by pressing a button. Sorry? Absolutely, you have. Now, that's a very good question because you'll only see the autosomal results, well, you used to be only able to see the autosomal results, but Family Tree DNA have recently introduced an X chromosome into their browser, so now you can also see your X matches, which is very, very helpful. Any other questions?
Hi, I was lucky enough to have my mother tested as well. So I have several matches that I match but don't match her. But my father has a very specific ethnicity. He's from a very tiny village. Often I find that my matches, there's just no chance that they'll match with my father, but they don't match my mother. So what is the chance that if they match me and don't match my mom, but I don't see how they can match my father. There was a false positive on my mom or something.
It's probably a very one of those segments that is very close to the threshold, because they won't report on segments that are less than 7.7 centimorgans with family tree DNA.
Yeah, but sometimes there'll be like 10 or 15 centimorgans that I'll match with someone. It won't match with my mom.
If you're matching, if you got a 15 centimorgan segment, then it's likely to be a true positive.
Yeah.
I mean, it still could have a false positive on one end and a false positive at another end. To get rid of those, you're down to 12 centimorgans. You could have that. But at that particular size of a segment, if you match somebody and your mother does not, it should be on your father's side. And he's done the family finder test as well.
No, he hasn't, and he won't.
Ah, well, okay. He would need to do the family finder test.
No, he won't.
He won't?
No.
Okay. If you match somebody and your mum has done the family finder test,
yes.
Well done.
Yeah.
Then the likelihood is that it is on your father's side. The way to confirm that, of course, would be to get your father to test, and you would find there in his matches the match that you match.
Yeah, I'm gonna chase my cousins on them, my paternal cousins. I think that's the
emotional
best.
blackmail works wonderfully.
Yeah. Thank you.
And we have a question here. Yes, there's the microphone.
If you get somebody a blood test, can you take a sample here without knowing something?
Well, the best thing to do is to sedate them. No. What we do over here is we do a swab, a cheek swab, and that's going to dislodge cheek cells, and we put it in a test tube off to the lab. You can extract DNA from hair, particularly hair follicles. The big problem is contamination, because somebody's going to have handled that hair. You know, we found it in the family Bible. It's probably great-great-grandmother's, but somebody has been handling it for generations, so you're going to get it contaminated. So the chances of actually extracting viable DNA and accurate DNA from a hair sample, I think, is less than 15%. So it's not a very viable way of checking for DNA. Like I said, just sedate them and then swab their cheek with you. Anybody else? Great. Oh, well, one last one. That's a strong voice you have there.
I'm projecting. I had a surprise in my autosomal analysis of ethnicity, 12% North African, and nothing in my. I've traced all my ancestors going back 5 generations, and there's no names or locations that kind of give a clue to that. What strategy would you suggest for trying to identify where that may have come from?
Well, if you upload your data to GEDmatch, there are a variety of other biogeographical analysis tools that you can use. The problem with biogeographical analysis, which measures your ethnic admixture, your ethnic makeup, is that they're only as good as the database to what they're comparing, to which they're comparing, and all of the companies have different databases. So I've tested with all 4 companies, and I have had different ethnicity results with all of them, to different degrees. With 23andMe, I am 99.8% European, 0.1% Sub-Saharan African, and 0.1% unknown, which I think is leprechaun. So it's going to be, and that is, they're refining it all in time. Ancestry have recently refined their biogeographical analysis. But what I would do is take it with a strong pinch of salt. You might be a completely different ethnicity next year. And try out those biogeographical tools on GEDmatch. You can also access them, I think, by doing a Google search for biogeographical tools or ethnic admixture tools. And failing that, just post somebody on the iSOG Facebook page, which everybody should join the iSOG Facebook group, and anybody there will be able to point you in the right direction. Great. Ladies and gentlemen, thank you very, very much for your attention.